My name is Frances Forrest. I am the manager of the Hall of Human Origins Educational Laboratory at the American Museum of Natural History. I'm also a zooarchaeologist, which is a fancy way of saying that I study mammals and other animal fossils that we find in association with human fossils at archaeological sites. And I work in northern Kenya in an area called Turkana. Very happy to be with you today.
Hi everyone, and I am Ashley Hammond. I am the Biological Anthropology Curator at the American Museum of Natural History. And I am also really excited that you guys have tuned in today. You're going to learn a bit about my research as well as work that Frances does in Kenya. I am a paleoanthropologist, so that means I study the fossil record for ape and human evolution. Most of my work that you'll learn about today is focused on the same region where Frances works, East Turkana in Kenya. So in just a second, we're going to transition to a video about my research, and you can learn a bit more about East Turkana and the fossils that are found there.
Fieldwork sometimes is not as glamorous as it might seem from movies. Fieldwork is often in a very remote place. In East Africa, for instance, it is very hot and dry, but it's a really magical environment. There's an element of discovery everywhere you go. You know that every time you cross that next little ridge or go down into the next gully, you might find a fossil. My name is Dr. Ashley Hammond, and I am the Assistant Curator of Biological Anthropology at the American Museum of Natural History. I am a paleoanthropologist, which means that I am a paleontologist who works on the fossil record for humans and our relatives. Everyone wants to find a hominin, a human ancestor. This involves field work, trying to get out to field sites that are at the very early time periods during which hominins would have evolved. My fieldwork over the last 5 years or so has really been focused in Kenya. The East African Rift Valley passes right through Kenya, and this allows a lot of old, ancient sediments to be uplifted and available for us to find new fossils. And the region that I work in Turkana, is especially great for this. It's really, in my mind, the most important place in the world for studying ape and human evolution. We have sediments that span the entire range during which apes and humans have evolved. The really best way to look for fossils at our sites is essentially walking across the landscape and looking for locations where fossils are coming out of the ground. So when we find something and we recognize that it's a hominin, typically the first thing I do is I tell everyone to stop and slowly back away. Maybe even, depending on the state of the fossil, take off their shoes, and then we can work the surface more delicately. We walk around or crawl around and flag all of the different pieces of bone on the surface. And then we go through and very carefully document and collect the fossil finds. So after we leave the field, we go back to the National Museum of Kenya in Nairobi, where the fossils are deposited for future generations to study. But I want to continue to work on hominins year-round. So what we often do at the museum is laser scan different fossils and then turn them into 3D models that we can work on here in the United States. So right now I am demonstrating our 3D scanner, scanning actually a monkey bone. And we can turn this monkey bone into a 3D rendering and use that as comparative data with some of our fossils. When you're working with fossils, often they are very fragmentary. So you have to tailor your analyses based on what you have to work with. You have to be more creative. So I developed some really advanced shape analysis techniques, things like fitting a sphere to a very fragmentary joint surface to estimate joint size. And this allows us to really compare shapes across different species or specimens in a much more advanced way than possible in the past. I'm really focused on trying to piece together the evolutionary history of the ape and hominin pelvis. The pelvis is really informative about the biology of an animal. It can tell you about what species you're looking at, whether or not you're looking at a male or a female. Sometimes you can even tell if an individual has given birth, and you can even get information about where an individual is from. I still think it's a really big question about what the starting point for the human lineage looked like, what kind of behaviors we were using, how we became bipedal, and so forth. So the only way to really get at that question is to get out and do more field work. So I'm very excited to get out with me and my crew and see what kind of fossils we can find.
All right, so we've got a few questions here from some of our viewers. So we have the Sandbox asks if we like our jobs, and the answer to that is absolutely. I love my job. So I love doing the research portion of my job and going out into the field and actually finding and collecting fossils, but I also just love talking to the public and sharing knowledge and research that people may not know about otherwise.
I agree. I absolutely love my job. I have a lot of similar responsibilities as Frances. I engage with the public and do research, but I also am a curator, so I also curate collections at the museum. So Frances and I are some of the main biological anthropologists that you would interact with if you came to the museum. So we hope all of you can come back when the museum reopens and visit us. Yeah. And next question, 8-year-old Ethan asks how we can tell how old fossils are. This is a really great question. So where Frances and I work in East Turkana, we are pretty fortunate that there is a well understood stratigraphy. Basically, there are layers in the sediments and there are ash layers in between different layers, and the ashes can be dated. So we know about how old different sediments are. And when you find fossils that are coming out of different layers, that then gives you brackets. Basically, you know how old or how young the fossil is likely to be. Now, it's not always the case that we find things that are coming directly out of sediment. Sometimes we find them sitting on the surface, and those situations become a bit more complex. We have to do a bit more detective work. There are also locations where it's not as easy to date the sediments. So some of the locations that are very famous historically for being difficult to date are places in cave sites. So for instance, in South Africa, it can be very difficult to date the sites, but we're making a lot of progress on that front too recently.
We have a great question from, I think it's Sweat Musk. So is there any evidence of direct competition between different early human species? This is an excellent question, actually. So as far as I know, we don't have any direct evidence. There have been theories of competition among or between modern humans and Neanderthals, because those are two species that actually coexisted with each other. Although again, we don't really have any real direct evidence of like physical competition. There's some partial evidence maybe that we were indirectly competing with them for access to resources. But as far as I know, I don't think we have any real direct evidence of that. Mm-hmm. Ashley, I don't know if you wanted to add.
No, I think that's very spot on. When it goes further back in the fossil record, there are some fossils that are found in the same place at about the same time that have very different adaptations in their anatomy. So that suggests to us that they were coexisting in the same place, but probably adapted for different lifestyles or different diets. So they weren't competing in the same way, but they were certainly on the same landscape and competing for resources. Mm-hmm. So I think we're getting the cue that we should switch to our next video.
We had one other question, I think, from Tristan. So how often do you guys go out on digs, if any? So I go out on digs less than Ashley does, but I usually go once every summer for about 2 months out of the summer.
And I go usually to 2 different field sites, both in Kenya for the last several years. I think every year I'm out for 2 to maybe even as much as 3 months per year in the field. Frances, you said about 2 months?
Yeah, around 2 months.
Yeah. So we spend a lot of time living in tents and getting field experiences in. Yeah.
Okay, so I'll introduce our next video. So this next video is talking about the past 7 million years of evolution. And so it's going to introduce you to a few different species of hominins or early humans. It talks a little bit about some of the important evolutionary changes that have taken place over the last 7 million years, and also touches on some of the relationships among these different species.
Okay, so you guys are getting a tour of some of the earliest potential hominins. So right now you're seeing Sahelanthropus shadensis. This is potentially the oldest hominin that we currently know about. It's about 7 million years old. And this is known primarily from a skull from Chad. Shortly thereafter in Kenya, at 6 million years, we have Auroran. This fossil has a very nicely preserved thigh bone or femur, which is suggestive of being an upright biped. So that suggests this could also have been a very early hominin. And then after that, we have a species called Australopithecus cadaba. And this is not a very well-known species, but there's a toe bone that suggests it was bipedal. And this species is related to a species that will come quite a bit later called Australopithecus ramidus, which we do know a lot about. And Australopithecus ramidus certainly looks like it was a biped. Now, the video is highlighting this chunk of time where we just don't have any fossil evidence yet for what was happening. So there is sort of a mysterious time period between about 4.5 and 5.5 million years ago. Okay, so now we've moved into Australopithecus ramidus. Australopithecus ramidus has a very interesting blend of somewhat more primitive features as well as things that we associate with the genus Australopithecus, which comes right after. So once you're in Australopithecus, you're clearly within the bipedal hominins. We have, especially from Australopithecus afarensis in Ethiopia, lots of fossils that suggest this was a well-adapted biped. And you can see early on, a lot of the fossil record that we have is really localized in East Africa. And this is in part just due to the geology and the uplift that we have in East Africa that's exposing the right sediments. Okay, so we're getting a few questions in. I'll try and answer these as we go. We have someone who asks, what's the difference between, Alec asks, what's the difference between hominid and hominin? So that's a good question. So I think historically you probably have heard people say hominid, and in recent years there's been a movement towards using hominin. So when we talk about the evolutionary relationships of some of these species, technically the word hominid has to include the great apes. So if we're being more specific, to refer to anything that is more closely related to humans than to chimpanzees, we would use hominin.
So we had a question from Mano, 7 years old. What kind of fossils have you found? So that's a great question. So lots and lots of extinct species. Mostly what we encounter are extinct antelopes, but we find fossils of all different sorts of creatures. So reptiles and elephants and hippos and rhinos. And of course, hominins, so early humans as well, although those are a lot less common than the other animals.
Probably less than 1% of what we find are hominins, unfortunately.
My favorite are the antelopes.
So we got a question from 6-year-old Olivia, and she asks, what did you study? What did you have to study to become a scientist? Well, everyone's path into science, I think, is a little different, and having a lot of diverse backgrounds is actually very, very beneficial to different types of science. Me personally, I started out in anthropology as an undergraduate, and then I got a PhD in anatomy, and that was really a great career choice for me because I want to understand how hominins were adapted, and I have to base all of my conclusions on their bony anatomy. All that we have of them is their anatomy. Frances, I think you took a slightly different career path. Is that right?
Slightly, yeah. I mean, so as an undergraduate, I studied biological anthropology. But then once I went into graduate school, I started focusing a little bit more on so animal anatomy, so less of the human anatomy and more of the anatomy of other mammals. So faunal analysis is what we call that.
Okay.
We have a good question here from Marilise. So why did early humans evolve to walk upright? What are they adapting to? This is an excellent question.
I wish we had the answer.
Yes.
This is something that I spend a lot of my time working on. We don't know why the earliest human ancestors initially began walking upright. There are many different ideas out there, and they range from perhaps changing environments and a need to either posture or move upright in a new kind of environment, perhaps feeding on different kinds of fruits or things that would have required them to stand upright. There's actually many, many different ideas about why the earliest ancestors would have stood upright. And at this point, we don't have enough fossil evidence to know yet. That's why it's very important for people like Frances and I to continue to go out every year and try and find more fossils. Because this question in particular is something that we're still working on.
Yep. So there are a lot of different hypotheses, but no real consensus. And then Anne asks, how do you tell what species a bone came from? This is a very good question as well. Lots and lots of practice. So just doing a lot of anatomical comparison between lots of different species and getting to know just little anatomical features that differ among the different species. And it just takes lots and lots of practice.
So I guess a fun question from Nancy is, what was your most exciting find? Do you have a most exciting find, Frances?
I do actually. So the most exciting thing that I've found was actually a pygmy hippo skull, and it had tooth punctures from a saber-toothed cat. And so we actually know how the individual died, and it was really nicely preserved. So I thought that was fascinating.
I remember that skull. It was the summer you found it. So my most exciting find, actually you were present for this find for me. Last year at a site I had been working for a couple of years, where some of the earliest evidence for Homo erectus has been found, I found part of a fossil pelvis for a hominin. And that was really exciting for me because a lot of my career has been spent on investigating adaptations in the pelvis in hominins. So I was very delighted to find that because it gives me an opportunity to work on not only the area that I've been focused on for quite a while, but trying to unravel what the earliest Homo erectus pelvis would have looked like.
How many of us watching might have some Neanderthal DNA? I feel like this changes periodically, but Ashley, do you know the most current estimate?
I don't know what the most current, the genomics field is a very fast-paced field. It changes almost on a weekly basis, it seems. But I think for the majority of people who are not from sub-Saharan Africa, the estimates put it somewhere around 2%, maybe 1 to 3%.
Well, there was a recent study showing that there is more of that ancestry in Africa than we had initially thought.
That's true.
So,
okay.
Marlene?
One last question, I think, is someone wants clarification on what bipedal means. So we should have probably explained that earlier. Bipedal means bi is 2, and pedal has to do with feet. So walking on 2 feet rather than walking on all 4s or being quadrupedal. Yeah, so being bipedal is one of the signatures. It's, as far as we can tell, the earliest adaptation that we see in hominins.
Yeah, and there are a lot of really obvious anatomical changes that come along with bipedal.
Okay, so now we are going to switch gears and go into a pre-recorded tour of the Hall of Human Origins here at the American Museum of Natural History. Feel free to continue to send us questions as you tour through the hall, and we will answer them once the hall tour is over. So we are at the American Museum of Natural History's Spitzer Hall of Human Origins, and today I am going to introduce you to a bit about paleoanthropology. So my name is Dr. Ashley Hammond, and I am the Curator of Biological Anthropology here at the museum, but I'm also a paleoanthropologist. So what that means is I study the fossil record for apes and humans. And so why don't you follow me, and I'll introduce you to some of our paleoanthropology here in the hall. So one of the questions that I always get from people is how we figure out how different things in the fossil record are related to each other. So what paleoanthropologists have to do is a little bit of detective work. So we typically compare skeletal material that we find in the fossil record to other fossils, for instance, Neanderthals, or we compare them to our skeleton or those of some of our closest living relatives, the great apes. And then we try and make hypotheses about how different things are related to one another. So I'm going to take you through sort of a chronological walk through time through some of the fossil record. We're going to start with some of the earliest hominins or human relatives that we know are related to us. So follow me, we're going to take a look at genus Australopithecus. Okay, so standing here beside me are 2 representatives of genus Australopithecus. Genus Australopithecus, so what is this? This is one of the earliest
clear hominin ancestors. So genus Australopithecus is comprised of a number of different species from places throughout East Africa and South Africa, and even now in Central Africa. We have fossils from all of these places for a number of different species, and they're all characterized by sort of a suite of characters. So genus Australopithecus typically are sort of smaller bodied, ape-like hominins. They typically have limb proportions that are more like apes, so they tend to have very long arms relative to their lower limb. They're bipedal and terrestrial, but they're also eating some pretty abrasive or tough foods. They have really thick enamel on their teeth. So they're different from us in a number of ways. You can also probably get a look here at the heads of Australopithecus. The cranial size is pretty small. It's really not much bigger than what we see in living species of great ape. Okay, so Australopithecus was like us in a number of ways, walking around bipedally, but certainly different from us as well. And we can tell that here.
And could you remind us of when we would find Australopithecus?
Sure. So, as I said, this is one of the earliest clear hominins that everyone agrees is a hominin. And we see genus Australopithecus from about 4.2 million years all the way back to about 2 million years. So a pretty long time period. Now, what you're looking at is Australopithecus afarensis. This is a species that is found in East Africa, places like Ethiopia and Tanzania. And this is actually a recreation of a very famous site called Laetoli, which is a footprint site where there were a pair of hominins walking side by side across the landscape. So I'm going to now walk you behind this and introduce you to the most famous of the Australopithecines, a little lady named Lucy.
And just before we go, we have one question from Michael Schiaparelli, who wants to know if primates would actually walk arm in arm like that, or if that was a creative license.
I think this is a little bit of a creative license. So what we know is that the footprints are side by side. We don't know if they were made exactly at the same moment in time. Perhaps one individual was walking a bit behind the other individual. But this is certainly how the artist chose to recreate this event. Okay, so this is the most famous of Australopithecines. This is Lucy. Lucy is also Australopithecus afarensis. And her skeleton was found in 1974 by a team led by Don Johansson. They were working in Hadar, Ethiopia. And Lucy just took the world by storm, because when we look at her anatomy, it's really clear that she is more like humans than she is like chimpanzees and apes. So let's get a closer look here. If we look at Lucy's pelvis, for instance, the way her pelvis flares out to the side is very characteristic of the human lineage. And we see some really radical changes to the pelvis that relate to being upright, being bipedal. And Lucy has those features already at 3.2 million years. We can also take a look at Lucy's thigh bone, the femur, and you can see that the thigh bone angles in towards the midline as you move down. And so this is what's called a bicondylar angle, and it's something that we really only see in hominins that are walking upright bipedally. And it allows us to more effectively transfer our weight when we're walking bipedally. We also see a number of other features in the skeleton that really make it clear that Lucy was a capable, competent, effective biped. But there are still some features in Lucy that look somewhat primitive. So some of her Lucy and some of her other compatriots in genus Australopithecus, they tend to have some primitive features. So for instance, sometimes their finger bones are a bit more curved. So this might reflect the fact that Lucy and her other sister species were still doing some climbing behaviors. We also see
kind of small joints. And so, especially, for instance, the vertebral bodies and the hip joint, places where there would have been quite a bit of weight transmitted during bipedal walking. So she wasn't as, derived as later hominins like genus Homo, but she certainly was already a very good biped. Now there are a number of other things that Lucy can tell us, though, in addition to the fact that she was a terrestrial biped. Okay, so what does Lucy's skeleton tell us? Well, we also know that Lucy was an adult. Her long bones no longer retain growth plates. They're fused, fully grown bones. So that means Lucy was not going to get really any bigger, any taller than she was. And that's really interesting because if you look at Lucy, she's really small. She's not just small for compared to modern humans. She was even very small for her species. So we know that she was not particularly large or husky, but certainly it also is really good evidence in favor that she would be a female. So in early species like Australopithecus afarensis, females were typically much smaller than males, almost half the size, in fact. And so Lucy was likely a female, and based on the elements that we have from Lucy, we've reconstructed her height, and she's really only about 3.5 feet tall. That's really small. So that is to put that in perspective, that's about the size of your average 4-year-old human. So she's not a very tall individual. Now,
we have a question from Matt Covery who wants to know, can you remind us how old Lucy is?
Sure, Lucy is about 3.2 million years old. Yeah. So pretty early on in human evolution. So now we're going to walk a little bit further into time, and we're going to take a look at something that looks a lot more like us in genus Homo. So we're going to go back here. This is another very famous skeleton, not as famous as Lucy, but this is an individual called the Turkana boy or the Nereokitomi boy. And this is an individual from genus Homo. It's an individual from what we call either Homo erectus, or some scientists also refer to it as Homo ergaster, essentially African Homo erectus. And this Homo erectus individual is so far the most complete Homo erectus individual that we have. And there's a lot of things that we can learn from this skeleton. First of all, those of you at home can probably already see that this individual looks a lot more like us and our skeleton than Lucy did. Okay, so if we look at the cranium, the skull, we can see that the brain size is a lot larger than we saw in Lucy and her species. So this is certainly not a cranium that would have been as large as ours, but it was on its way to becoming more human-sized. We also see elements of the skeleton that really look a lot more like humans. So for instance, if we look at the limb proportions, we see that this species looks more like our own. So the lower limb, the leg bones, are longer than we see in the upper limb. And this is a reverse compared to what we see in apes, and it's a reverse compared to what we saw in Australopithecus. So that suggests to us scientists that this was a species that was really completely focused on being a terrestrial biped. This was not a species that really would have exploited the trees at all. And this was a very effective biped, walking about probably pretty far distances. Now, what's also really exciting when we enter genus Homo is we start to see other signs that link the genus with us. Okay, so we start to see more advanced behavioral complexity. Okay, and this reflects that the cognitive abilities have really increased in our genus. And so once we're in genus Homo, we start to see better evidence in the archaeological record for things like advanced tool use and meat eating and potentially even hunting pretty early on. And so if we look at this diorama here, you get a little glimpse of that. This is another artist reconstruction of a hominin species. In this case, this is a reconstruction of Homo erectus. And you can see that we have two Homo erectus individuals here that are scavenging a bovid from the African savanna, and they're fighting off some predators, a jackal and a vulture. They're also using tools. But you can see that, this is a species that, although much more like our own, they're still different from us in a number of ways. So they're not yet producing clothing or using really advanced toolkits yet. So do we have any questions from the audience at home?
And we've had some people commenting on how old things are.
Okay, so that's another really exciting thing about Homo erectus. So Homo erectus is a really long-lived species. Okay, so we looked at Australopithecus. Genus Australopithecus, I told you, was about 4.2 million to about 2 million years. Homo starts to appear around 2.8 million years, but Homo erectus, this particular species, appears in the fossil record about 1.9 million years. And it actually, the earliest evidence for Homo erectus appears in the region where I do field work in Kenya, in a place called East Turkana. So we have very early evidence at about 1.9 million years for this species. And then very soon after, we see evidence in a number of other places, even in Eurasia. And Homo erectus seems to stay on the landscape until perhaps as late as 30,000 years ago. So that's a really long period of time. That's almost 2 million years of this species in duration. And if you think about that, our species has only been around for a little over 200,000 years or so. So Homo erectus was a species that was around 10 times longer than us so far. Of course, we don't know how long the human species will sort of be on the landscape, but certainly Homo erectus, although it's extinct now, was a very successful species for a very long time.
We have a few more questions, but maybe could you take a step into the light a little bit there? There we go. We have a question from Dan Opperthauser, who wants to know, why are there so few fossils of these species?
Well, so I think that's a little bit of a misconception, actually. So there are lots of fragments of these species. It's hard to find skeletons due to the nature of how the fossil record works, but there are actually, at this point, thousands of hominin fossils that we have from various places around the world. So we have quite a bit of fossil record and evidence for the shape of how human evolution happened. It's the time period that is really, really patchy is sort of around the time period when the chimpanzee and hominin lineages would split, so about 6 million years ago. But after about 4 million years, we have a really, really strong fossil record.
We have a question from Claire Houlihan, who wants to know, do you think the climate condition in Kenya is a factor in preserving these bones?
So it's a number of things in Kenya, and there are different kinds of environments that preserve fossils in different ways. So one of the things that you need pretty much regardless of what kind of environment is you have to have water. So it typically requires water and water moving sediments on top of a skeleton to produce a fossil. So in Kenya and East Africa generally, they are part of the East African rift system, which is exposing the new fossils today as the continent rifts apart. But in the past, this kind of tectonic activity also played a role, and it created lake basins and river patterns that would have picked up skeletons and deposited them and led to their fossilization. And so the location where I work in northern Kenya around Lake Turkana had a large lake 4 million years ago and forward called Lake Lorenyang. It's roughly in the same position as where Lake Turkana today is. And this ancient paleo lake is what allowed so many fossils to be deposited in this one place. Now that differs from other places. So for instance, there are a lot of hominin fossils that are recovered from cave sites. And this is true in places like Spain at a site called Atapuerco, where there's a large number of hominins recovered in a single cave. And we also see this a lot in South Africa, where there are cave systems that hominins get sort of washed into or potentially even deposited by carnivores. And we actually have a great exhibit of one of these right here. So what you're looking at is a cave floor deposit from a site in South Africa called Sterkfontein. And I'm a little sentimental about this one because this is the first hominin that I ever got to see in person about 15 years ago. I went down into the cave to look at the fossil as it was in the cave deposits. And this is a cave system that the sediments become rock hard, like concrete. It's something called breccia. And there are skeletons or bits of skeleton that are preserved in that sediment. And so this actually, in the last few months, this skeleton has been partially published. So it's now out of this sediment and breccia, and it's becoming available for study. But this is an example of what you might find in certain cave deposits, where you have sort of a large amount of bones that have accumulated. And generally, cave deposits tend to be better at preserving whole skeletons as compared to places such as in East Africa.
We have a question from Jean Hart, who wants to know if we have estimations of population for these different species.
That's tough. There are people who work on modeling population sizes for extinct hominin species, but these models are often based on modern human parameters and modeling. So it's hard to say. But in certain places, such as these cave sites, where you might end up with multiple individuals, and potentially even from a single population, you can get a sense for approximate number of individuals. You can get a sense also for the composition of the group, about how many adults versus how many children there might be. And so there's really only a few places where this is possible. So we can look at these kinds of questions at, for instance, a site in South Africa called the Rising Star Cave System, where it seems that there's a number of individuals possibly from the same population that have been preserved. We can also do something similar at a site in Ethiopia where it's called the First Family was preserved. So there's a family of Australopithecus afarensis that was sort of catastrophically killed in a single event, probably a flood event. And then we also can do something kind of similar to this at some of the European cave sites where there seem to be multiple individuals. But it's very hard to estimate population sizes and population composition unless you really clearly have a population that died in a single event.
We have another question from Pamela Ruby Cajudo Navarez, who wants to know if hominin fossils are rock fossils, I guess in the way that dinosaur fossils we might think of are.
Yes. So the process of fossilization is essentially that the soft tissues and collagen in the bone is replaced by other minerals. And essentially the bones are turning into rock. Now, there is sort of a time period in the fossilization process where things are sub-fossilized, where they're not fully fossilized. And sometimes those fossils still are a little bit soft or pliable. They're not fully rock yet. And the more recent fossils are really exciting for other reasons, which we haven't covered. And that is the more recent fossils, if they're preserved in the correct conditions, can sometimes produce DNA and proteins. And then we can work on reconstructing sort of the evolutionary relationships, not just based on the anatomy, which is what we mostly do, especially for the old fossils, but we can start to look at relationships based on molecular data, based on DNA. But there are a number of things that limit when we can extract DNA. So there is a time limit. DNA degrades. So right now we have only extracted DNA from things that are 400,000 years or more recent. So right now we really only have DNA for things like Neanderthals and a very mysterious species called the Denisovan hominins. Another thing that limits DNA preservation is the climate. So DNA tends to degrade faster in hot climates. So of course, the fossils in Africa are typically in hotter and sun-baked localities. So the DNA is not likely to preserve in those places. But sites, especially in Europe, that are permafrost sites or cold weather sites will preserve DNA pretty well.
And Joao Silva wants to know how archaeologists come up with the names for the different specimens.
It depends on the different teams. So often we decide names. There's typically a genus which has already been established, and the genus names also often have some significance behind them in terms of the Latin roots of the word. But what we see now is species names are often selected to honor the location or the local people for where the hominins are found. So we often see words that have a root that is not Latin, but one of the local languages or locations.
I want to thank you so much for taking the time to show us around today. Really appreciate it.
All right. So Ashley, do you want to talk about the cast versus real fossils? We had a question about this.
I did see a couple questions about whether or not the fossils on display are the originals or if they are replicas. So at the American Museum of Natural History, we only have casts on display, and that is intentional. The real fossils are part of the cultural heritage of the countries in which they were found. So fossils that were discovered in Ethiopia remain in the National Museum of Ethiopia. Fossils that were found in Kenya remain at the National Museum of Kenya, and so forth. So this is an important thing for the home countries to retain the access and control of the fossils from their country.
So there's a question from Lennon. What inspired you to be an archaeologist? So actually, this is an interesting question because I had absolutely no idea what I wanted to do when I started college. And I just started taking a whole bunch of different classes, and I ended up taking a class in human evolution and just thought it was the most fascinating thing I had ever learned about. And then had a chance to actually do some field work in Kenya, and then I was hooked after that.
And my story is similar, although different. I knew from a young age that I was very interested in anthropology. And I think I wasn't sure once I got into college what the next steps were. And I just took as many anthropology courses as I could, and eventually was able to go out into the field. And just like Frances, I felt the field work sort of transformed my life. And there was never any question after that about what I wanted to do. So we had a question from Adriana about, she says, we have visited the National Museum of Natural History because we live close to DC. Our kids want to know if your museum work is together with the NMH or if we share information and collaborate. The answer is yes. So collaboration is a very important part of science, and not just for scientists directly, but for museums. There are a lot of collaborations that occur between museums. We will share exhibits sometimes. And even when it comes to research expertise, for instance, although I work at the American Museum, I still retain affiliations with the Smithsonian in order to more easily collaborate with researchers there. So I think the answer is yes, we do try to facilitate as much collaboration as possible between the scientists and the museums directly.
So Laura asks, what can we do to become part of your team? It's amazing what you guys do. So I think the short answer is, I guess, a lot of school. So we had to go through undergraduate and then master's and then PhD programs. Although I can give a little bit of a plug, Ashley and I work for a field school where we take undergraduates out with us to the field. So if you know somebody who is an undergraduate student that would be interested in coming out into the field and just seeing what it's like, they would be able to come with us to Turkana.
Yes, and in addition to our field school, there are many, many other field schools out there that explore different time periods or regions of the world. So there's lots of field opportunities out there for interested undergraduate students. So we had another question. Do we know what the chimp ancestor right after the split with humans look like? And they further elaborate and say chimps must have evolved over the past 7 million years too, correct? This is a fantastic question, and I am really excited that someone answered this because this is something that not many people think about. So the split between the human lineage and chimpanzees happened sometime between 6 and 8 million years ago, let's say 7 million years. That means that not only have hominins been evolving for 7 million years, but chimpanzees have, their lineage has also been evolving in some way for about 7 million years. So we don't know exactly what the ancestor of chimps and humans look like. We don't know if it looked more chimpanzee-like. There's just simply not enough evidence. And unfortunately, this split occurred during a time period where we don't have many fossils. So this is why I said earlier, it's really important for people like Frances and I to get out and continue to do field work, because there are these time periods that are having really critical events in human evolution that are poorly sampled. So the short answer to the question is, we don't know what the ancestor of chimps look like, but we shouldn't assume that they looked exactly like chimpanzees.
There's another very good question. What's the biggest mystery in human evolution that you would like to see solved? So I think my answer may not be as interesting to people as it is to me, but I'm actually really interested in early human diets. And so one of the things that I would like to know more about is the incorporation of meat into the diet. And so if that was something that was a very important transition in human evolution, is it something that was a survival strategy initially and then became more of a common practice? How those early humans would have had access to meat? Were they hunting? Were they scavenging? So those are the sorts of questions that I'm really interested in.
And I think for me, the biggest mystery in human evolution that I'd like to see solved actually relates to the last question that I answered. I would really like to know what the ancestor of the chimpanzee and human lineage look like, what both groups evolved from. To me, that's the biggest unsolved mystery, that we just don't have any fossil data on that yet.
All right. And do we have time for this last one?
Yep.
So are there scans or 3D models of these fossils anywhere? And how do you all share information? What does it mean to publish a fossil? So there actually are sites where you can get 3D models or 3D scans. Some of these are private and some of them are actually accessible to the public. So I think Morphobank, is that one actually open to, accessible to the public?
Morphosource.
Morphosource, right.
That is publicly accessible. And then There are several others. So for instance, africanfossils.org also has fossils online, and they can be, as Frances said, easily downloaded. And there's free software out there that allows you to manipulate scans pretty easily to take a look at the anatomy in the fossils. And if you are even a school teacher or someone who has access to some of these 3D printers, you can 3D print some of the scans relatively easily. And then the final part of their question is, how do you share information and what does it mean to publish a fossil? So I think the most important aspect of sharing information for scientists typically is some sort of publication, some published record of what you found. If you don't share your results and your data in some kind of publication, there's no way to ensure that it will exist in the public sphere in perpetuity. So that is probably the most important final end product.